Okay, ladies and gentlemen, we're going to talk bacteria today. Scientists think that bacteria are the oldest life form on this planet. They think bacteria arrived about 3.5 billion years ago, whereas the planet's age is estimated to be between 4 and 4.5 billion years. In fact, they even think that mitochondria and chloroplasts actually were once free-floating bacteria that moved into eukaryotic cells. Mitochondria and chloroplasts seem to have their own DNA, and they seem to divide separately from the rest of the cell. And so that's a reason to believe that maybe they had been bacteria at one time. They're also similar in size to bacteria, and so that's more evidence toward the idea that mitochondria and chloroplasts were once bacteria. Now, there are so many different kinds of bacteria, and they're so unique from one another that science has actually divided them into two domains or kingdoms. The Eubacteria are the ones that we come in contact with, and a lot of people refer to them as germs, although very few of them actually make us sick. The Archaebacteria are more ancient. They're found in unusual or extreme environments. I'll mention them for just a few minutes here, and then the rest of the time we'll spend talking about the Eubacteria. All right, so if you can quiz yourself here, what evidence supports the hypothesis that mitochondria were once free-living bacteria? See if you can come up with that answer. I'm not going to answer it for you. You can pause this. Video at any time to go back and take a look at that question. See if you can find the answers. And where do the Archaebacteria live? What do we call the kind of place where you find the Archaebacteria? All right, moving on. Examples of Archaebacteria. There's three types that we're going to talk about. The first are the methanogens. Methanogens are anaerobic, which literally means without oxygen. And they release methane, and they are found in places like swamps, they are found in sewage, and they're found in the intestinal tracts of animals. Unfortunately, the methanogens are found in the intestines of cows, and when cows fart, they release methane, and we are learning now that cows are a huge producer of global warming gases. Methane is one of the worst global warming gases, and apparently a lot of it has to do with agriculture and cow farts, of all things. So those are the methanogens. That's one kind of Archaebacteria. The next kind of Archaebacteria we're going to talk about are the extreme halophiles. Hallow refers to salt, and phil means, as you know, loving. So these are salt-loving bacteria. They use salt to make their ATP. And these tend to be found in places like the Great Salt Lake and the Dead Sea. And then the last category of archae or ancient bacteria that we're going to talk about are the Thermoacidifies. You would find Thermoacidifies in a place like Yellowstone National Park. They live in hot, acidic environments, as you can tell from the name. So they're found in places like hot springs, volcanic vents at the bottom of the ocean, and cracks in the ocean's floor. Now, the rest of the time we're going to spend talking about the Eubacteria. The Eubacteria are bacteria that you are much more likely to come in contact with. They live all around us, on our bodies, in the soil, on the things that you touch. These are the Eubacteria. The good news is that less than 1% of bacteria are actually harmful to humans. Most bacteria don't care about us at all, and many of them are actually helpful to us. Some help us digest food or make vitamins. Some help us fight off disease-causing bacteria. And some are even used to make foods like yogurt and cheese. The eubacterial structure is what we're going to study at this point. The eubacteria has something called a capsule. Oops, I didn't mean to do that. Let me try that again. Sorry. There we go. It has something called a capsule that protects the inside of the bacteria, and it helps it to stick to surfaces. The capsule is not to be confused with the outside of a virus, which is called a capsid. They also have a cell wall, but the cell wall is different than a plant cell wall. The plant cell wall is made of cellulose, whereas the bacteria cell wall is made of a giant molecule called peptidoglycan. So it has a cell wall, but it's not like a plant cell wall. It has DNA, just like you and I have DNA, but the DNA is circular. We often think of our chromosomes as X-shaped. They're not really, but the DNA of a bacteria is circular, and the bacteria has no nucleus. In fact, a bacteria is probably smaller than the entire bacteria is probably smaller than our nucleus in any of our cells. It also has pili, which are kind of like hairs that help the bacteria attach to surfaces, and they're hollow, and DNA can be traded through the pili, so you can see them there on the diagram. So what percent, as a question here to quiz yourself, what percent of bacteria make humans sick? See if you can remember that. And describe bacterial DNA. Is there a nucleus to store the DNA? Things you should be getting out of this as we go. All right, here's an interesting question. Soap works by making surfaces slippery, allowing sticky bacteria to slide off. And soap works without any additives. Soap is very effective. Recently, however, soap companies have begun adding antibiotics to their soap because they think that consumers think antibiotics in their soaps make their soaps better. Considering the risk of increasing antibiotic resistance, some people want this practice outlawed. Should soap companies continue this practice? Why or why not? Do you think we should be adding antibiotics to our soaps, or do you think it should be outlawed so that they cannot use antibiotics? We'll talk more about antibiotics in a few minutes. All right, so how do bacteria reproduce? They reproduce basically through mitosis, but when it's a bacteria, it's called binary fission. So it basically clones themselves. They make identical copies of themselves. So then the question becomes, how do you get how do you get bacteria that are different? And that depends on some things that we're going to talk about in a little bit. While bacteria don't have sex, they can exchange DNA with other bacteria. So there's 3 ways that we're going to talk about that. A way that a bacteria can get new DNA, the first way is called transformation. This is when one bacteria is actually able to suck up the DNA or the plasmid of another dead bacteria. And so now that new bacteria, that bacteria that sucked up the DNA from a dead bacteria now has new DNA, and that causes that bacteria to change. That's called transformation. In conjugation, if you've ever heard of a conjugal visit in prison, it refers to prisoners being allowed to have sex with their married partners. Conjugation refers to bacteria exchanging DNA through their pili. To call it sex is not very realistic because there are not male and female. There's no parts that make them male and female. But they do trade DNA through those pili, and it's a way of getting fresh DNA. And so it allows bacteria to change, to be modified. The third way is a little bit unusual. It's called transduction. This is when bacterial viruses, remember how we talked about bacteriophages? That's a bacteriophage sitting on a bacteria right now. It is possible for bacteriophages to pick up the DNA from one bacteria and transfer it to another. And then we have new DNA added to a bacteria. So that's another way that bacteria can change or mutate. So how is it that bacteria make us sick? Bacteria release poisons, poisons that can do several damaging things to us. They can kill our cells or our tissues. They can paralyze the cell machinery inside so that our cells aren't working properly. And they can cause an overreaction of the immune system, which might cause things like an excess of fluids in the lungs or something like that. So that's how bacteria tend to make us sick. So they also, sorry, I didn't say that very well, they may reproduce so quickly that they crowd out healthy tissue and disrupt normal functions. So how do we fight bacteria? This is a very different part than how we fight viruses, because we have antibiotics. Viruses are totally immune to antibiotics. Antibiotics work by interfering with bacterial cell functions. They interfere with how cell walls are made, for example. So we have these medications that we can take. And I just want you to take a look at the diagram because it's super interesting. These are 4 petri dishes that have food on them, food that bacteria would like to eat, and there's bacteria growing, and each of the bacteria is a different color. So they've been stained with these different kinds of bacteria. And then if you notice, there are these little discs. They look like little round paper punches. Each of those round paper punches has been treated with a different antibiotic. This is a way that, for example, a lab technician at a medical clinic might be able to test an individual for antibiotic resistance or what the best antibiotic a doctor should prescribe. So what might happen is a doctor might swab a patient's throat or their nasal passage and then smear the bacteria on this petri dish. And then they add these little antibiotic discs to the petri dish and allow the petri dish to grow for 24 to 48 hours. Anywhere that the bacteria does not grow indicates that the antibiotic in that area is effective at killing that particular bacteria. So wherever you see a clear area around one of the antibiotic discs, that's a useful or effective antibiotic. But anywhere that you see a disc that has been completely grown over, that's an antibiotic that bacteria already is resistant to, and if the doctor prescribed that antibiotic, it would not be effective. So anywhere where there's a big clear circle, that's what you would want to prescribe for the antibiotic. So here you have an example. Which antibiotic would you prescribe? Take a look at your choices there. Which one would make the most sense? All right, so what exactly is antibiotic resistance? Where does it come from? So this is how antibiotic resistance occurs. A group of bacteria is exposed to an antibiotic, and the susceptible bacteria die. That means basically the weak bacteria die. What does that leave behind? That leaves behind any bacteria that are mutant who are resistant to that antibiotic. Usually those mutant bacteria are killed by the immune system. So there's just a few. Most of the bacteria are killed by the antibiotic, and there's just a few mutants left behind that your immune system can capture and kill. However, there are many people in our society who have weak immune systems. The elderly, the very young, AIDS patients, people on immunosuppressant drugs, etc. And they might not be able to fight off those last few strong or mutant bacteria. They might have a hard time fighting off those bacteria. So then those mutant bacteria start to reproduce, and they produce a colony of resistant bacteria. So now that you've used the antibiotic, and if their immune system doesn't kill those last remaining ones, those are the ones that reproduce and produce a whole huge colony of antibiotic resistant bacteria. So then what do? Now a new antibiotic has to be used, and the process can repeat itself. Unfortunately, there are some bacteria in the world that are now resistant to all known antibiotics. There are some types of tuberculosis, which is a very serious and deadly lung disease. That's something like 1 in 6 people around the world have had exposure to tuberculosis. I don't remember the exact numbers, but and there are some varieties for which there are no treatments anymore because all antibiotics the bacteria are resistant now to all of their antibiotics. So how can we prevent antibiotic resistance? Well, first of all, you would only want to take antibiotics when you have a bacterial infection. Most colds are caused by viruses. Sometimes apparently people go to the doctor and they really want the doctor to prescribe them an antibiotic, and the doctor feels pressured to give an antibiotic so they feel like they're doing something. But if the illness is caused by a virus, an antibiotic does no good. That said, when you do have an antibiotic prescription, when you do have a bacterial infection, it is important to completely finish the prescription even if you are feeling better. If you destroy all of the susceptible bacteria with antibiotics, your immune system is free to focus on the resistant bacteria. If you stop taking the antibiotic prematurely, there may be some other bacteria still there or more bacteria still there, so the immune system is busy taking care of them and can't focus on the mutant or resistant bacteria, and so you're more likely to get another bacterial infection. They're more likely to be able to evade your immune system, so to speak. And that's it.